Well, great. It's so nice to see everyone virtually today for our single-cell spatial analysis program seminar series. For those of you who don't know me, I'm Justin Colosino. I'm faculty in the School of Public Health, and I'm one of the PIs on this University of Michigan Biosciences Initiative supported program. So today we're lucky to be joined by a star in the spatial oncology space, Dr. Alastair Lamb. So Dr. Lamb is a cancer research UK clinician scientist at the Nuffield Department of Surgical Sciences at the University of Oxford. And as you'll see today, Dr. Lamb has been doing excellent work at the forefront of using spatial analyses to map clonal somatic mutations and expression profiles within aggressive tumors. And I really think that this work is an outstanding example of the translational power of these spatial omics methods to impact patient care. And we're really, really excited to learn more today. So we'll wait until the end of the lecture for Q&A, but if you have questions, feel free to add them to the chat. Then after the lecture, we'll open the floor for questions. So without further ado, I will turn it over to Dr. Lamb. Dr. Lamb, thank you so much for joining us today. We really appreciate it.
No, that's an absolute pleasure, and thank you for having me. And am I allowed to ask one or two quick questions? In terms of, I can see the lecture theatre obviously looks empty, but I can see lots of people joining online, which is exciting. Thanks for that generous introduction. Are we joined here by mainly basic scientists, biologists? Do we have a mixture of clinician scientists, biologists?
I'm like looking through our current attendees, and it looks like we have sort of like basic scientists, clinician scientists, and maybe some clinicians.
Wonderful. Thanks for giving me that little.
Oh yeah. So for your question towards the auditorium, we've been trying for these remote lectures. We've been trying to set up little like satellite areas on campus for people to watch, but it looks like most people are watching from their offices or at home instead.
Totally understandable. The convenience of virtual meetings in the post-COVID era. Great. Okay. Shall I share a screen?
Yes, please.
I'm going to share sound because they've got a couple of short video clips. And you don't want to see that.
Looks great.
Okay, so hopefully you've got a front page.
Looks good to me. Yeah.
So, oh, thanks, Justin, for the introduction. As you've heard, my name's Alastair Lamb. Yeah, I'm a cancer biologist and a urologist, a prostate surgeon. That is, I investigate prostate cancer as a disease biologically, but also treat it and operate on it surgically, diagnose it as a clinician. And I'm really looking forward to sharing with you some of what we've been up to in the spatial genomics space, trying to unpack particular issues relating to heterogeneity, disease heterogeneity in prostate cancer, using novel platforms to interrogate the spatial landscape of this interesting disease. So yeah, I'm speaking to you from Oxford. As you can see from the window, just over to my left, it is dark. I imagine your evenings are drawing in as well, but I know it's the middle of the day for you. But yeah, we may not have sunsets like that top strip at the moment. Certainly in the summer we do. These two buildings at the bottom, the Churchill Hospital on the left is where I do my clinical work. That's just where I've been today. I actually just finished an operating list doing robotic prostatectomies today. And over on the other side is our old road campus research building, which interestingly is actually also where the AstraZeneca vaccine, the Jenner Institute, is situated. And you'll have obviously heard about that on the radio waves over the past couple of years. So thanks very much for having me. So just a brief overview, and actually before I jump in with that, I just want to say I'm not sure if I haven't had the opportunity to visit Michigan in the past. I'd love to have it in the future, but we have just recently sort of formalized a collaboration with one of your colleagues there, I think Todd Morgan. I'm not sure if he's joining us on the call on the meeting, but and a very excellent DPhil PhD student called Sophia Abisamra has joined us in Oxford with joint supervision from myself and a couple of others, and input from Todd. So it's exciting to have that connection as well with you guys in Michigan. Okay, so here's the overview. What we're going to cover. I'm going to give you a bit of background to prostate cancer. why it's such an interesting disease. I think of it almost like the rainbow cancer. It's lots of multiple different colors, and with the sort of spatial maps that we use, that's particularly relevant. I'm going to talk you through the SPACE study, which is my research program, as a prelude to our paper, our organ scale paper, we call it, looking at clonal dynamics. If time, I may touch on some surgical aspects, and then give you some future directions arising from that. So prostate cancer is extremely common. As you can see here, it's the second highest in terms of the share of the world's population with cancer. To put it another way, I sometimes end up having conversations at dinner parties, and a fellow guest may ask how likely they are to get prostate cancer. And I tell them that if they're the owner of a Y chromosome, that they have a roughly 50-50 chance of getting prostate cancer if they live to the age of 80. How likely are they to know about it? Well, actually, that's about 10%. I'll put up some Venn diagrams here. And how likely are they to die from it? Well, that's much less, at 3%. And herein lies the problem with prostate cancer. It's very common, but the vast majority of men with this disease will never know about it, let alone die from it. Although, of course, 3% of all men is still a lot of people. And there's another way of outlining this problem, which is the huge range in survival rates with prostate cancer. It can be as lethal as, say, bladder cancer, or indeed kidney cancer, lymphoma at the left-hand end of the spectrum, this wide, wide bar. Or it can be one of, if I may say it, the best cancers to have in terms of survival over on the right. We tend to risk stratify prostate cancer using a combination of three things: a non-invasive biomarker, the PSA test, which you get from a blood test, TNM staging, which is normally imaging or clinical examination based, tumor node metastases, and then biopsy or histology, the Gleason score, still one of the most accurate predictors of outcome. But there are big inconsistencies here with lots of crossover between risk groups. Some men who we expect to do well doing poorly, and vice versa. So let me tell you about our program of work, the SPACE study in Oxford, which I was funded by Cancer Research UK for a 5-year program, and under the oversight of my excellent biologist colleague Ian Mills and my mentor and head of department Freddie Hamdy, another prostate cancer surgeon and neurologist. So the SPACE study, spatial prostate assessment and the circulating environment. And our context is high-risk localized prostate cancer, which we interrogate using a number of techniques: biopsy, surgical removal, image-guided sampling, and blood sampling. Our goal is to identify the lethal clone. And there are three components to what we do. Stage one is looking at MRI-guided biopsy of the prostate to enable interrogated genomic analysis at the first sampling point in prostate cancer. Second, we seek to unpack clonal heterogeneity across the whole prostate and augmented from lymph nodes and other circulatory sources the small number of cancer cells which have left the primary lesion, so-called micrometastatic disease, to enable us to identify this lethal red clone over here on the right, the one which potentially at the time of radical curative treatment has already escaped the prostate. And then to circle right back, if we can, to identify this lethal red clone at the time of the first sampling point in prostate cancer. And this is really important. And the spatial analyses that we've developed and seek to employ here are critical, because here's a fantastic paper which my lab, where I did my PhD in Cambridge under mentorship and supervision of David Neal, published back in 2015. Really exciting piece of work called the Complex Men Study, analyzing prostate cancer, but in bulk at multiple sites, and demonstrating indeed the heterogeneity of this disease, but with great limitations delivered by the fact that it was only a bulk analysis at these different sampling points. So really pushing the boat out, but still limited and largely guesswork in between. So to summarize so far, our objective is to unpack the clonal dynamics, the heterogeneity within localized prostate cancer, and to employ various other approaches in the circulatory system and analysis of lymph nodes, circulating tumor cells, etc., to try to identify which of these clones have escaped this red clone that I mentioned in the previous diagram, or orange perhaps here, which at the time of radical treatment is already in circulation, therefore potentially the likely source of lethality and recurrence. So this is what I think you wanted me to talk about after I've given you a bit of background, which is our organ scale paper recently published, which my excellent recently departed postdoc Andrew Eriksson was the lead author on, along with Meng Shao and Emily from Joachim Lindeberg's group. There's been much to be sad about in the COVID pandemic, but I suspect many of us were aware of certain silver linings. And for my part, I can certainly say that this project is one such silver lining. In 2019, Joachim kindly hosted Andrew over at his lab and at the SciLife lab to get really his head around spatial transcriptomics, which I knew was going to be a really critical platform for that second stage, if you like, of our 3 stages of the space study. Up to that point, people have been sort of digging in deeper and deeper into the sort of resolution question, but I felt we needed to take a step back and look at the whole prostate because of this multifocal heterogeneity that we've talked about. And we were on the cusp in beginning of 2020 of applying spatial transcriptomics to whole organs. And then COVID hit, and Oxford took quite a draconian approach and closed down all patient recruitment, unless you were working on a COVID-related study or with AstraZeneca. And so, yeah, basically because Oxford placed all their eggs in that basket. But this was a problem for the rest of us. So it was such a relief when Joachim reached out to say that they had some whole prostates from some colleagues in Stockholm and had recently done some Visium spatial transcriptomics and had a piece of work they were keen to collaborate with us on. And could we help with some of our ideas and the computational elements? It was perfect timing. We basically then spent a couple of years working on this together up to this paper coming out in the summer or in August. So what have we done here? Well, I think infrastructure in cancer is extremely important. For too long, we've been ignoring the spatial infrastructure, the spatial context of both benign epithelia particularly, and the transition to cancer. And it's because we often forget to focus on campus infrastructure that we actually know very little about the transition from benign to malignant tissue. What actually happens in this process? Not helped, of course, by the fact that when we have tried to analyze this, we've bulk sequenced to get often very detailed genomic analyses and lovely strong genomic readouts, but we've got no idea often where they come from. And as I've mentioned, this is really important in prostate cancer because of this spatial heterogeneity, as demonstrated in the paper that I already mentioned. So because of our focus on cancer, perhaps rather than benign tissue, because of our limitations with bulk sequencing, we've often overlooked benign and the transition from benign to tumor. And our objective in this piece of work was to study the spatial genomic integrity in situ and derive clonal relationships from that. So here's how we go about this work. We particularly focusing on the prostate, but there are other tissues, and I can touch on those as we go through it. The schematic shows how we take a cross-sectional disc of prostate, a bit like in that Nature Genetics paper you just saw, and we divide it into cubes and take 10 micron sections for spatial transcriptomics, which I'll come to in a moment, but as many of you will know, uses a spatially barcoded approach. I suspect lots of people are now reasonably familiar with this. Here we go. In brief, this is 10x Genomics Visium version 1. So 6 millimeter squares with barcoded spatial probes and with a poly-T tail intended to capture the poly-A tail of RNA fragments on the tissue. Now, this is a 3' read with approximately 150 to 200 base pairs in length. So it's not the whole gene sequence. And this is, we've found, obviously, totally suitable for mapping to the human genome for transcript readouts, and as you'll see, for copy number alterations as well. Question is whether it can be used for other genomic inferences, such as SNVs, which we feel it might be in that limited area, possibly rearrangements, we're not so sure. So our approach is contingent upon 3 important phases. We have our observed, scientifically observed, using the platform Spatial Transcriptomics. And this technology was one which, as you may know, Joachim Lindeberg and his group invented, and a very nice paper in 2016 from Patrick Stahl and the rest of the team. We used the single cell derived spatial, or infer CNV, sorry, and derived from that our spatial infer CNV, which is taking that sort of baseline from the Patel paper from the Broad Institute back in 2014 and applying it in a spatial context, as you'll see in a moment. And then the third layer, and this is really key, is really granular, detailed consensus pathology underlying all this. So across this, the different tissues in this paper, there are over 150,000 spatial infer CNV measurements. And of those, over 50,000 spots within that have a consensus pathology readout, essentially treating each spot like as a pathology call, where at least 2 pathologists here, my colleague Richard Colling in Oxford, and a great friend and a collaborator, Thomas Murti from Helsinki in Finland. A lot of work on the pathology side, and we believe that's really important. So I want to move on now to some validatory steps to why we think we've managed to derive effective infer CNV or copy number events from our transcript readouts in the spatial transcriptomics. And one way to do that, of course, is to take a monoclonal cancer, in this case, a pediatric brain cancer, medulloblastoma, which we can then bulk sequence. So just to give you a brief outline here, the heat map on the right is an infer CNV heat map. Each row represents a spot, as shown on the left. And the reason they're all red is they're all one clone. And you can see here this chromosome 3p gain and 9q and 3q gain and then 9p gain and 9q loss. And because of the monoclonal nature of this tissue, we were able to then validate that using whole genome sequencing. We were really pleased to see this because this validated our infer CNV approach. So if we accept that we can use inferred copy number alone to track clones, then let's progress to the whole organ. And this is what we do. So basically we assign each spot. This is obviously a cartoon, much magnified, but each spot is 55 microns. I'll show you what that corresponds to in terms of the tissue level in a moment. And we use that to assign spots to clones and then undertake clonal phylogenetics, as you would hope we would. And let's move to the organ scale level with the prostate. This is a whole axial section of the prostate divided into square cubes. And we sort of generated this schematic, which I think is quite a nice summary of the histological composition of each of those cubes. 2 dominant areas of cancer, one large one over on the right here with, this is Gleason grade group 4 and grade group 4 cribriform, so aggressive disease, with some slightly lower grade grade group 2 next to it, as over on the left-hand side here, and then a small area of low risk or low grade cancer, Gleason grade group 1. And let's take a look under the hood here, as you say in the US, to see what's going on. We'll select one section, shall we? That one, H25, the top part, sort of on the edge of cancer and benign epithelia. And you can see here, cancer, this slightly unusually named transition state. You can ask me that later if you want, but that's what the pathologist wanted to call that. This is really unusual, highly granular pathology here. So they had to come up with some quite specific ways to call these spots, different to as you would with the standard pathology report for a prostatectomy specimen, for example. And then if we look at one small region here, we can see here the kind of resolution that we're working to. So here we've got an area of cancer in red, with the red spots, an area of benign epithelium. And what they were doing here is requiring spots to have more than 50% of a particular epithelial type, be that cancer epithelium, benign epithelium, or indeed stroma. And if that wasn't the case, then these spots, as in black, for example, were excluded, either because of a mixture or because stroma or benign epithelium were, sorry, a mixture of cancer or benign or stroma and epithelia. So very granular pathology. I'm just going to show you as a quick aside now, very short, just over a minute video. It's just kind of outlines as a summary of what I've talked about so far and gives you a pointer to what we're going to talk about next. Sound should be shared. Justin, if you just maybe give me a shout out, the sound doesn't work, but I think it will.
With new spatial transcriptomic technology, we can now analyse both the genetics and the spatial landscape of tissue. We've sampled multiple sites in several organs and inferred their genetic status from their transcriptome. Rather than bulk analysing each tissue section for one genetic readout, we instead get several thousand spot sequences per sample, each to a resolution of around 10 cells, and we can allocate each spot to a clone, allowing us to create a phylogenetic tree that parallels the transition from benign to malignant tissue. We found multiple subclones within an area of tissue, and surprisingly, we found genetic changes thought unique to cancer are also found in seemingly healthy tissue.
So I'm going to just focus on one or two aspects of the story, but I hope that you'll see from those aspects the sort of the power and, to be honest, the potential for further work as well, and potentially for collaboration as well. So let me start by explaining how we create our reference set. So we wanted, the way that infer CNV is you can apply it without any reference, but if you have a reference set and you define that as your sort of diploid set of subspots, then it allows you to accurately call copy number gain and loss in the areas of interest. So here what we've done in one prostate is we've taken all of the histologically, with consensus pathology, defined spots as benign, histologically defined benign spots, and allocated them to an infer CNV heat map. And we're probably talking 45,000 spots here down the y-axis. Now, this is interesting because, as you can see here, there's an area that is not diploid, has some copy number gains and losses. And that was fascinating. We'll come back to that in a moment. But we selected here this region as our sort of pure benign, if that makes sense. Okay, so this is now our reference set as we move forwards. And let's then have a look at what's going on with those alterations at the bottom. Now, interestingly, they all seem to be located in one section here, H21 over on that left-hand side, which is an area of tissue containing both cancer, some prostatic intraepithelial neoplasia, putative pre-malignant area, although that's a little controversial, and benign tissue alongside stroma. But we're just literally focusing on the epithelium here, not including the stroma. Most of the Gleason grey group 2 cancer, small area grey group 4. Well, if we then allocate each of these spots to a spatial infer CNV heat map, very briefly, we're taking the infer CNV approach and requiring spots to be present in spatial proximity with at least 10 spots allocated to the same clone. So in other words, we're enriching for similarity and difference. And this is similar to the previous heat map, but we're focusing on just spots in this section, each spot again being a row working down the y-axis, and obviously chromosomes along the x-axis. And I think what you can see here is, while there is some heterogeneity moving down through the spots, there's also a lot of homogeneity within what we've called these clonal sub-areas. And let's see where these, so moving from A through to G, let's see where these are located. So as you can see on the left-hand side now with the overlay, so we are requiring them to be spatially approximate. They have to be at least 10 spots in one location to be called a clone. But you can see, interestingly, that this basically follows the branching morphogenetic pattern. So this is a ductal epithelium moving down from up here, if you can see my arrow in the green area, through the yellow and into orange on one side and red on the other side. And I'd like just to focus on one region here, which is the yellow clone C, which actually has a lot of copy number gains and losses, broadly similar to these lower clones E, F, and G. And that yellow area is located here in an area of histologically benign. And, you know, the pathologists, we really asked them to double check and triple check this, and they are histologically benign. So not all, but the majority of the copy number events we normally consider to be pathognomonic of prostate cancer are actually already present in this clone C, which is histologically benign. That's fascinating. And of course, what you can do when you have this granular level of detail, and when you can see also the histological structure, is you can produce a phylogenetic tree. I'm very grateful to my colleague in Oxford, Dan Woodcock, for his extensive advice on these. And that's demonstrated over on the right there. Now, some of you may still doubt whether our spatial infer CNV is accurate, really is calling copy number gains and losses. So we focused on two, I highlighted them on the previous heat map, two particular oncogenes, one oncogene and one tumor suppressor, c-Myc on chromosome 8q and the PTEN tumor suppressor gene on chromosome 10, which you saw in the previous heat map. And we validated our spatial infer CNV calls using fluorescence in situ hybridization, and we were really pleased to see that using these squares, true benign, altered benign, and cancer, that we were able to see what we were expecting to see based on the spatial infer CNV. Thanks to Reza from Joachim Lindeberg's group for that. So yeah, we were really pleased to see this, and it gave us confidence in our calls. I mentioned the Gleason Gray Group 1 area, and that's really interesting, because this is a type of prostate cancer that clinically many of us are fairly confident now doesn't progress to higher grade disease and can't spread. I mean, there are one or two case reports, but almost the fact of their existence, and everyone knows what they are. Michael Hafner's paper from Baltimore being one a couple of years ago, which was cited as the example of Gleason Gray Group 1 metastasizing. But basically, we don't think it does. And it's increasingly affecting how we manage this low-grade type of cancer. And some are, like Matt Kuperberg in San Francisco, are calling for it to be declassified as cancer, or histologically it is. So let's have a look at that little area of yellow. And we applied the same sort of approach, spatial infer CNV analysis, to these areas. Interestingly, so that's the area of Gleason Gray Group 1 there, at higher resolution. There are two clones within that. But interestingly, what you'll see is they lack many of these changes that we saw, for example, 8p loss, 8q gain, 10p loss. So those are just a couple of examples that I've mentioned, and which we validated with fluorescence in situ hybridization. But low-grade prostate cancer seems to lack those. There are other events present, whereas that altered benign area of epithelium did contain those areas. So, does that mean, for example, in terms of the clinical applications of this, that if we were to detect an area of benign but copy number altered epithelium, that has a more lethal phenotype, potentially risky phenotype in terms of diagnosis and then prognosis than low-grade prostate cancer? That's an interesting question. A couple of other validations. I said that we found that we could take the short sequence at the 3' end and undertake SNV analysis. So we inferred SNVs in the short region of that exome available to us, and we're able to confirm using variant allele fractions that this was indeed allele-specific inheritance going down through those clones in the subclone analysis. We also conducted an in silico modeling experiment to seed cells in a fictional 2D tissue with stochastic acquisition of copy number events on a single chromosome in both the poly and subclonal fashion. We did make certain assumptions about cis-associated mRNA levels and overlaid our fictional ST grid and spots to generate an siCNV profile. And pleasingly, this almost exactly recapitulated the underlying copy number ground truth in this in silico experiment. So for more detail, please do have a look at the paper. So I'm going to finish with some future directions, what we think we can use this work for going forwards, and hopefully give food for thought for some ideas and share some of what we're hoping to do next. I've got a short video to finish. So our focus, as I said, is on both intratumoral heterogeneity and extraprostatic clonal seeding as a window into lethality. And so far, as you've seen, we've really been focusing on that left-hand side. In order to work out which of these clones within the prostate, which we now feel we're able to identify, track, interrogate, but in order to identify which of those really matter, we need to know, we need insight into which of those are spreading into either the lymphatic or blood circulation to form lymph node metastases and bone metastases, the two common sites in prostate cancer. So here's one thing that we're doing. Don't worry about the details here. This is kind of showing you our working. This is our sort of mock-up that we make. This is a recent prostate that we've taken from the operating theatre. Apologies to those who are eating lunch, as I realise it's lunchtime for you. That's a prostate in the middle. We identify the location of areas of cancer from the MRI scan and use that when we create this axial disc that you've seen to localize certain areas of cancer and look at areas of, for example, local advancement into the seminal vesicle or the capsule of the prostate. That's the sort of first stage of a cancer spreading in prostate cancer. Another area that we're working on, and this is actually now using the Visium version 2 protocol, which allows us to use bigger squares, which we're really keen on, and also to work with FFPE. This is just a mock-up. We don't have this data yet, but I'm just sharing. It's not secret. This is what we're doing, but sharing where we're heading in the future. So here's a multifocal prostate cancer, and what we do is we divide it into squares. Take these discs, these squares at 11 millimeters in width, and we're working with that at a Visium version 2 level to try to track the spatial location of clones within the prostate as they spread to lymph nodes. And of course, the idea here would be then work out which of those clones depart either in local advancement or regional metastases, and what is the relationship between further clonal spread between lymph nodes, within lymph nodes, or indeed further afield. This is a project which I've briefly alluded to already when I mentioned Sophia, who's come over and joined us from Michigan just in the last couple of months. And so the idea here is to use circulating tumor cells as a window into lethality. So there's a lot of work to be done here, which is, Sophia's excitedly getting her teeth into. And under the joint supervision of my colleagues here in Oxford, Jason Davis, who's a chemist, Claire Edwards, bone metastasis biologist, and Ian Mills, who I've mentioned already. And at the moment, we're still in phase 1. We're using a magnetic field-activated nanoparticle approach, and we'll be excited to see whether we can use that to identify circulating tumor cells, initially in metastatic disease, which is not controversial, but in localized high-risk disease as well, as I mentioned, as a window into lethality. Here's another area of what we think is low-hanging fruit for this work. Many of you will know that prostate cancer is diagnosed using core biopsies, not whole gland analysis. And so we had this idea that we could reconstruct a virtual biopsy using our spatial transcriptomic data. And the purpose of this being to demonstrate that the disease heterogeneity within the prostate means that it does matter which bit of the prostate you sample. So let's take a couple of virtual, putative tumor biopsies, biopsy 1, biopsy 2, and we compared them to a fictional benign biopsy. This is an idea that Olivier Cousineau, who's a urologist and scientist from Paris, spending a year with us as a sabbatical, came up with. And what you see if you then look across those reconstructed fictional biopsies, histology at the top for both biopsies, is that it's really heterogeneous. It really matters. This is just transcripts. This is not inverse CNV here, just transcripts. It really matters which bit of tissue you look at. And by and large, these biopsies, when they're sent off for genomic tests, Decipher, Polaris, Oncotype DX, are either analyzed in bulk or just the tumor is microdissected and the benign areas left behind, but the tumor is then analyzed in bulk, ignoring the different grade group subtypes or indeed clonal subtypes. And so we've been trying to use heat maps and GSEA plots to demonstrate this and provide some sort of scoring system. It's quite hard to unpack the black box of how these genomic scores is applied, but I think the point stands that heterogeneity in prostate cancer matters. So Sandy had this idea, and this is where the rainbow idea came from, that it would be interesting to look at proximity to an area of tumor. Again, something that's not been possible in the past before the spatial transcriptomic approach, but we now think could be. So here's your rainbow. Let's analyze transcript changes as you move from close to further away from a large area of cancer. What effect does that have on prostate epithelia, on areas of stroma? Is there crosstalk between these areas? Perhaps cancer-associated fibroblast impacts on the epithelium in terms of transformative change, phenotype, that kind of thing. Another thing we're interested in is the timing of the branching events in copy number, using copy number as a clonal window into somatic mosaicism. And so this was just a mock-up that we made as a supplementary figure in the paper, tracking those branching morphogenetic changes that I mentioned. But the question is, if these are present in non-transformed, histologically benign areas of epithelium, when did that happen? In the testosterone surges back in utero? Probably not. In puberty? Possibly. In later life? Perhaps in a more slowly developing acquisition of mutational change. We don't know. Interesting project to study. Multiplexing, apologies for the quite a bit of detail on this slide, but we're interested to look at combinations of different platforms, PhenoCycler for protein, spatial transcriptomics we talked about, and possibly using light sheet microscopy at a three-dimensional histological level as well. And this is what it's all about. I've come back to this slide a few times, but we're trying to marry up intratumoral heterogeneity in the prostate and the lethal clone. So here's a video to finish, and then I guess we'll have time for some questions. This is just 4 minutes long, and I'll finish with this.
There's been a real challenge in prostate cancer to better understand precisely what's going on within a prostate. We've come up with a new way of analysing prostate cancer tissue, which should allow us to work out which cancer cells spread and which don't. This study is a joint collaboration between ourselves at Oxford University and Joachim Lundeberg's group at SciLife Lab in Sweden. One of the main things we've discovered is that many of the genetic changes that we previously thought were uniquely associated with cancer tissue are actually already present in seemingly healthy prostate tissue.
This is particularly important because we usually think of cancer being an entirely different entity to healthy tissue, but cancer has to start from somewhere, and identifying these areas within the same region of a patient's biopsy, we can identify the place where the cancer has grown or areas that have the potential to become cancer themselves.
We We also found that areas of cancer that look the same actually have quite different genetic composition, suggesting that they arise from different parent clones, the cells where the initial mutations occur that drive cancer.
In order to get human tissue through so many different locations, even in close proximity in Oxford, is quite a difficult task.
Our spatial transcriptomic pipeline allows us to take tissue from the operating theatre to the lab so that we can undertake our unique new analysis.
To do all of this pipeline, we have to use different techniques, as cryo-sectioning the tissues of fresh frozen tissue, extracting DNA information from that tissue, and preparing sample for sequencing. It's really important to have different expertise as data analysis, biology, clinician.
We do have a very rich data set now that we can interrogate to move away from just the genetic background.
We have a fantastic critical mass of prostate researchers here in Oxford, which have been drawn together over the last few years by Freddie Hamdy, our head of department. And this is alongside our awesome clinical team, our urology department at the Churchill Hospital.
The work that goes on behind the scenes in order to develop the research does not only span different expertise within the same team, but it spans different institutes within Oxford, and in our particular case also different institutes across Europe.
We came together in the early part of COVID when we were all getting good at Zoom meetings. Joachim's group had access to large amounts of this new technology because they invented it and wanted to work with a really unpredictable cancer like prostate cancer. And we realized we could help each other with access to data and tissue. I see this as a bit of a silver lining of COVID, actually. There are currently many men who we treat who probably don't need treatment. These treatments have side effects, be that incontinence, erectile dysfunction, or menopausal type symptoms because of hormone therapy. Being able to unpack what's going on in a man's prostate with real accuracy. Is this cancer clone one that's primed to spread, or may it just sit there and never bother you? This is key to treatment in prostate cancer. It will allow us to give the right treatments to the right patients, so that if possible they can avoid the side effects of treatment. Patients really care about this, and their families do as well. We think this work and the ideas arising from it could revolutionize how we treat a number of cancers, such as breast cancer, skin cancer, and brain cancer. And we are really looking forward to collaborating with others who might wish to be involved in taking these ideas forwards. We're really excited to see what comes next.
There we go. I think, to be honest, I think the summary is there in the video. I don't need to go through that. I'm really grateful to my team in Oxford, many of whom you've seen in that video, actually. And I mentioned Andrew, who wasn't able to come over from Finland, sadly, Andrew Eriksson. And I'm also super grateful to Joachim Lundeberg's group, without whom we wouldn't have been able to make this project happen. And he continues to be a great collaborator. So thank you very much. I will unshare.
So thanks, Dr. Lamb, for an outstanding talk. It was really good to see those really nice videos, good scientific communication, and as to why this is such an important area. So as folks are formulating their questions, you can feel free to put them in the chat or raise your virtual hand to ask, and maybe I'll take moderator privilege to ask first. I guess I have two questions. So one is easy but nitty gritty question is, and that's how many reads per spot would you recommend for reliable CNV data if you want to regenerate this type of data?
Yeah, that's a really interesting question, because actually for the FFPE, we've been a bit concerned as to whether we'll get enough signal to be able to call these accurately. I mean, look, 50,000 reads per spot is what we aim for, what we allow for, and I think we get that with the frozen tissue. So of course, and then 5,000 spots per section, and you can do the maths as to how many therefore you're looking, how many reads you're looking to get when you sequence these samples. It's a lot. So look, I think that's worked for us. 50,000 per spot at a copy number level. Obviously, it's not deep sequencing, is it, per spot if you're looking for more than that, and you are limited, of course, to that 200 base per sequence.
Great, thanks. That's really helpful. And then I guess I have a more sort of like bigger picture question. So somebody that's like really interested in carcinogenesis and trying to understand, like what makes tissues cancerous. So it was very intriguing to observe these like regions of benign that have the cancer like CNVs. And maybe this is what Sandy's project was getting at, and maybe you don't have an answer quite yet, but have you been able to mine the spatial transcriptomic data to figure out if there's like something in the microenvironment that's like keeping those clones in check, or kind of like what's going on there?
Yeah, it's fascinating, isn't it? Why don't those copy number events, which we think are, as I said, pathognomonic of cancer, why if present are that clone not transformed? I don't have the answer to that yet, but of course we're looking at that. I think it was a contact resolution, so we're really interested to look at the stromal compartment and stromal epithelial crosstalk. One thing we've noticed, however, is that our pathologists, who are wonderfully skilled at calling epithelial histology and subtypes, find it harder to call the subtypes in stroma, be that in myofibroblasts, smooth muscle, etc., inflammatory infiltrate. And so we think probably we're going to need to use a computational approach in terms of deconvoluting, because there is still deconvolution required here, because we're still looking at 10 cells per spot, maybe 15 cells per spot.
Sure. I think maybe there's a question.
But I'm really
go ahead.
intrigued in that. I think that'll be a really intriguing line of investigation to answer that question about interactions, microenvironment.
Yeah, I think that was super interesting. It seems like y'all have like the great data to do it. And then I think there's a question in the chat that's related to kind of what you just said from Andreas Patsalos. So it's they say, thank you for the nice talk, Dr. Lamb. Taking into account the limited resolution of the technology, so like 55 microns per spot, I was curious if you're familiar with Bayes space. This is a fully Bayesian statistical method that uses information from spatial neighborhoods for resolution enhancement into subspots. Have you tried it on your spatial data to enhance your clustering or plan to? And if so, what do you think about it?
No, I haven't, although I know a bit about Bayesian analyses from my clinical trials, interestingly. I've taken a screenshot, and I'm going to chat to the team about that. So thank you so much.
Yeah, sounds like a nice approach. Yeah, some of our colleagues here at U of M are thinking about ways we can do the sort of like the tissue level deconvolution, and it sounds like Bayes space might be a nice way to do it. Are there any other questions? So I'm not seeing any pop up in the chat. So I think, were there any questions in person at Palmer over there? I see a couple of people showed up.
No, we don't have any questions over here.
Okay. That's great. So I think we all really appreciate your time and just sharing your just fantastic work in this area. I think we're really excited about seeing these next steps too. I think looking at the circulating tumor cells in relationship to the prostate cancer, I think it's just going to be so insightful as to understand really the origins of metastasis, and there's a lot of power here. So I think thank you so much for taking the time and spending your evening with us here. We really appreciate it. Hopefully we can get you here in Ann Arbor sometime soon.
I'd love to. Just send the invitation.
Great. Thanks so much, Alastair.
Joking aside, it'd be a pleasure, and yeah, it'd be nice to for an opportunity to foster some collaborations.
Yeah, I think there would be a fruitful ground there. Yeah. So thanks so much, everybody. We'll see you next time.
Thanks, Justin. Thanks, everyone. Thanks for having me.
Thank you.